All right, so now let's move on to the cytoskeleton. The cytoskeleton is a meshwork of fine fibers in the cytoplasm of a eukaryotic cell. Its main function is for structural support, just like our main function of our internal skeleton is for structural support. So this is just a model here where they're highlighting these different filaments. So this is showing these filaments in here. Now there are three types of filaments that we have that make up our cytoskeleton. The first are called microfilaments, and that's this little guy right here is a microfilament. So microfilaments are the smallest filaments. They're going to help change the shape of the cell and also help that cell to contract. Next are intermediate filaments. So this is showing an intermediate filament right there. They're called that because they're intermediate in size. And they're going to help anchor organelles, so holding those organelles in place. Next are microtubules. So microtubules, literally small tubes, but they are the largest of the three filaments. They're going to help provide cell shape. They're also going to provide tracks for organelle movement, so these are the larger ones that you see here. And they also are going to move the chromosomes during mitosis, all right, or cell division. So this is another picture of those. All right, so let's move on here to the centrioles. So there are the centrioles, and you're seeing microtubules come off of those centrioles there. So what the centrioles do is they're going to guide the movement of the chromosomes during cell division. So they essentially are going to control those microtubules. All right, so this is just a blob of those centrioles there. So if we move to this picture now, let's talk about appendages. So these are things that stick off of the cell. Now, why I'm listing them underneath the cytoskeleton is because they're made of microtubules. So the first are cilia. So this is cilia right here. If we go to this next picture, we can see cilia there. All right, so here is cilia here on the edge of this protus. So cilia, these are short, numerous, hair-like appendages. And when we talk about a single-celled organism like this guy here, the cilia there they move that organism. All right. Now, when we talk about a larger multicellular organism, we're going to find these. They line our respiratory tract. So in our nasal cavity, they're in our sinuses, they're in our trachea. And here, what they do is they're going to move substances. In this case, they're going to move mucus. Okay? So when we inhale air, we don't just inhale that air, we inhale anything that's dissolved in that air. Dust, dirt, pollen, bacteria, viruses, whatever is there. And so we produce mucus to help clean that air before that air gets to our lungs. And so through ciliary action, we are going to move that mucus. So if it's in our nasal cavity or sinuses, we're going to move that mucus down to our throat, our pharynx, and then we swallow that mucus and it goes down to our stomach, and our stomach acid is going to kill pretty much whatever's in there. All right? If it's our trachea or windpipe, ciliary action is going to move that mucus up to our throat, and then, I just swallowed some more mucus again. All right, and then that's going to go down to my stomach. My stomach acid is going to take care of that. Now, obviously, you don't taste this mucus, but I do want to point out, like anytime you swallow, you're swallowing mucus that is drained up from your upper respiratory tract or being pushed up through ciliary action to your pharynx from your lower respiratory tract all the time. Okay, so now let's move on to cell modifications of the cell surfaces. And the first one I want to talk about are cell walls. So this is a rigid boundary surrounding a cell. So we see this with plant cells, we see this with fungi, and a lot of algae have different kinds of cell walls. In fact, we're talking about this, most life has cell walls. Animals don't, and there's some protists that don't as well. Now, their cell walls are made of different things. So plants and a lot of algae, their cell walls are made out of cellulose. With fungi, it's a combination of proteins and carbohydrates. And then when we talk about protists in general that have cell walls, there's a large variation that can occur there. They could be made out of cellulose, they could be fungal-like in that protein carbohydrate compound, they could be glass, they can be calcium carbonate, which is what chalk is made out of, so that's made of a bunch of dead little organisms. All right. So what cell walls do is they impart shape and also regulate the cell volume and also protect the cell. All right. So one of the problems though with cell walls is they don't allow substances to move from one cell to the next. So plants have these little holes called plasmodesmata. So these are essentially channels between adjacent plant cells, and they allow substances to move from one cell to the next. So now, nutrients and waste can move back and forth between those cells. All right, let's take a look at animal cells. Now, animal cells are held together by a sticky outer layer made of glycoproteins, and that's what we see out here is what that is stuff. All right, so there's other things that are going to hold cells together. So the first are what are known as tight junctions. So these things right here are trying to show tight junctions. This is an area of adjacent plasma membranes where the cells are fused together. All right, so we have proteins on either plasma membrane. Those proteins are going to stick together. And this forms a leak-proof sheet. And this little dotted line is trying to show nothing can go in between those cells. And this is good because we have these along cells that line our digestive tract. So that way, the food that we eat is here, and it can't move in between those cells. And that's a good thing because we release enzymes in here that break down carbohydrates and proteins and lipids. So then we take in the broken down products of those and we absorb those into those cells. Okay? So that also prevents those enzymes from moving in between there, because those enzymes wouldn't be good for us either. All right? Next are desmosomes. We see desmosomes right here. This is a cell junction composed of thickened plasma membranes that are joined by filaments. All right? And so what desmosomes do is they hold these cells together. All right? So this is found in cardiac muscle cells. It's found on skin cells as well. Lastly are gap junctions. So down here are gap junctions. We see those right there as well. This essentially is a passageway between two adjacent animal cells. So two animal cells close together. It's essentially like a large straw that, allows substances to move from one cell to the next. What's nice about this is that if we have a blood supply over here, this cell can get oxygen and nutrients from that blood supply, and then through these gap junctions they can move to the next cell, and then through these gap junctions they can move to the next cell. And then waste products can move back in the opposite direction. So this makes it so not every cell in our body has to be directly connected to a blood supply. All right, so let's switch topics here. So the last topic of this chapter deals with the endosymbiont theory. So the endosymbiont theory states that the eukaryotic cell evolved from a larger prokaryotic cell that engulfed a once free-living bacteria. So what endosymbiont means? So an endosymbiont is an organism that can only live within another organism. All right, can't live outside of it. So there are two parts to the endosymbiont theory. All right, so it's basically how we got these eukaryotic cells and how they got some of the organelles in them. So two parts. One is the origin of the nucleus and the endomembrane system. So I think this is the pictures out of this book. I like this one out of a different book. So here we starting off with a prokaryotic cell, and we see these the plasma membrane has folded inward. And we can find bacteria that look like this, and they have what we call respiratory membranes on them. So when we look at the origin of the nucleus and endomembrane system, it's come from this growing inward of the plasma membrane, and then over time, this became the nucleus and the endomembrane system. Okay? So this added more surface area, so you can have more reactions occurring. And here's the thing about this. We can find bacteria that look like this, and we can find eukaryotic cells that look like this today. All right? Next is the origin of the energy-related organelle. So I'll come back to this picture. And so what this states is that the mitochondria and the chloroplasts were originally bacteria that were engulfed by a larger cell. Okay? And so that's what we're seeing in both of these cases here. So, and this is what we see here as well. So let's look at evidences for this. So both chloroplasts and mitochondria are similar in size and shape to bacteria. All right, so that's one evidence. Next is both have RNA that is similar to bacterial RNA. All right, both have their own ribosomes. All right, so they can make their own proteins. Both divide by splitting like bacteria, and they'll do this separately from the cell. So this is the odd thing, right? When our cell is going to divide into two cells, we kind of double up on our organelles. That's not necessarily the case with mitochondria and chloroplasts. Both are double membrane. So this is one of the things I want to mention here. They're double membrane. So the inner membrane is the plasma membrane of the bacterium, and the outer membrane is the plasma membrane of the larger cell, the engulfing cell. And so that's how these guys became double membrane. And lastly, something I've already mentioned to you as well, is both mitochondria and chloroplasts contain their own DNA. Now, when we studied this DNA, chloroplasts DNA is more similar to cyanobacteria, the photosynthetic bacteria, than it is to any other organisms. All right? And also mitochondria, we can put mitochondria in a specific genus of bacteria called Rickettsia. So that's how similar it is to other bacteria that's out there. All right? And anywhere along this line, we can find organisms. So we find bacteria that are like this today, bacteria that are like this today, no chloroplasts or mitochondria in them. All right? And so what had to happen here is that aerobic bacterium became the mitochondrion, and this then led into the fungal line and the animal line, and then this line where they engulfed the chloroplast became the plants and the algae.